For our fourth video, we're going to be going over genetics, DNA, proteins, and how this all contributes to Friedreich's ataxia. We talked a little bit about this in our first video, but I want to go into a little bit more depth now for those who might want to have some more information on it, and so that we have some more background when we talk about research and the hope for a cure down the line. In order to do this, I'm going to do a tiny bit of review to just mention that our DNA functions as a blueprint in our body, creating proteins, and these proteins serve different functions. Our body has tens of thousands of different types of genes, all making different types of proteins, all helping the myriad of different types of functions that our body does. In our case, we're most interested in the gene called FXN, which creates the protein called frataxin. All right, well, let's back up a little bit. What are proteins made of? They're made of different types of amino acids, which are molecules that our body uses as building blocks in order to make up these proteins, so that our proteins can have many different shapes and structures to perform the various functions that they have. So now we know that our FXN gene assembles different amino acids in order to make our frataxin protein. Well, if there's an error in both of our copies of DNA, it won't be able to properly create that protein. But how do we get from a whole bunch of list of molecules that we have in our DNA, billions of molecules long, as a code in order to actually make that protein? Well, every group of three molecules that are in that DNA encode for a different type of amino acid. There's some more exceptions that go along with this, but for our purposes, it's good enough to know that every time there's a group of three molecules within our DNA, it tells us to add a different type of amino acid to the protein we want to make. We can create a whole chain of these in order to tell us what our eventual protein is going to be. Our DNA just uses pairs of A and T, which we're going to use as shorthand for two different types of molecules, and C and G as pairs in order to tell us what information it is, and ultimately what amino acids we're going to have. Think of this really as just kind of like binary code, like zeros and ones. I'm not a programmer, I couldn't program anything to save my life, but ultimately we know that computers use codes kind of like we speak on written language or write things down in order to give information. Likewise, our DNA uses codes of these molecules and pairs in order to tell our body what amino acids to make what proteins. So when we have our gene like FXN, we're going to have all these groups of three molecules together that are going to tell us our code in order to have our amino acids in order to create our protein. All right, so now we have this background of what it should normally work like. For our next part, let's look at what actually happens with the FXN gene and frataxin in the case of Friedreich's ataxia. Continuing on with our discussion of how we go from DNA as a blueprint in our cells in order to make protein, we're going to learn a little bit more about what goes wrong and what happens in Friedreich's ataxia. In order to do that, we have to at least learn a little bit about the steps in the process. The whole entirety of the steps wouldn't really fit on the board and is kind of beyond the scope of our discussion today, but we're going to learn the most important ones so that we have an understanding of what goes wrong and how we might potentially be able to fix that down the line. All right. So our DNA is a chain of billions of molecules long with thousands of different genes in it. We're only really concerned about that FXN gene for now. That FXN gene has a really long code. I'm not going to write all of it out, but essentially it's a whole long list of molecules that creates that code for our body to decide to make the protein. All right, so we have this long code, which I'm going to represent here. That code needs to get trimmed and shaped. The body has actually a longer list of coding in that gene than is really necessary to make the whole protein because it uses pieces that it clips out in order to shape it appropriately. Different segments of that code are called introns and exons. In the process of making the code ready to make the protein, we actually snip out the introns using all these different little helper molecules that come in and help shape it appropriately. All right. So we get to that trimming and shaping stage, and then we get to the short code, which is actually going to turn into our protein. Once we have our short code, we remember that every group of three in it tells us a different amino acid that's going to form our ultimate protein. This can be thousands of amino acids long in various cases, but our body and our cells takes this shorter code to put all these amino acids together, string them together, and shape them into our ultimate protein. Proteins are folded in all kinds of weird shapes in order to perform their various function, but if everything works appropriately, we go from our long code, take out the pieces that we don't need so that we can shape it the right way, end up with our shorter code, use that as a blueprint to add the amino acids we want and fold them into our correct protein. Next, we're going to look at what exactly goes wrong in the process. So in the case of Friedreich's ataxia, instead of having this appropriate trimming and taking out of the introns in our code, leaving only these exon parts of the code to form the correct protein instructions, unfortunately, there's a big repeat of parts of the code that we don't want. As we mentioned before, there's 4 different types of molecules that make up the code of our DNA: G's, A's, C's, and T's. Well, in Friedreich's ataxia, unfortunately, there's way more of these G's and A's in this repeating pattern going GAA, GAA, over and over again, about 90 to even up to 1,300 different times. This all happens in this one section of this first intron in that code that we normally want to have snipped out in order to form our normal function. Unfortunately, with Friedreich's ataxia, people have this extended code in both copies of their DNA, and this really long part of the protein that's normally supposed to be taken out doesn't work as well. You still have all the little different helper molecules that come and try and cut things into the correct form so that we can eventually go on and use our shortened code to form our protein. But the reality is that this larger and misshapen code doesn't work as well as it should, and it's harder to get our shorter code and to turn that into our correct protein. As a result, you end up with far less of that frataxin protein that you need for normal function. In fact, depending on the person, you typically end up with only 5 to roughly 30% of the normal protein that you're supposed to have. One of the things that's important to note is that the length of these extra repeated molecules that you have in your code is ultimately has a big impact on how much of the normal protein you make. The longer this repeat is, unfortunately, the less of that protein that you're going to create normally. That's part of why you see a genetic counselor, and they can give you an idea of how long that repeated code is and how likely you are to have more severe symptoms from the disease. Approximately 96% of patients who have Friedreich's ataxia have this particular problem with the repeating GAA in both copies of that FXN gene, which causes their Friedreich's ataxia. There are some exceptions to this and other issues that can happen with the DNA code that cause the Friedreich's ataxia, but fundamentally they all work in the same way of interrupting the process of making the proper frataxin protein, which would help create the normal function that that protein's supposed to do. We're now going to take a look at why Friedreich's ataxia is known as a recessive genetic disease. Recessive genetic diseases are ones where you require 2 copies of an abnormal gene in order to have symptoms, whereas a dominant genetic disease would be you only need to have 1 copy of the abnormal in order to have any symptoms. We're going to ignore dominant for now and just focus on recessive because that's what's relevant in this case. Let's take a look at this particular example where we have a person with 2 copies of the gene, just like everybody else, one from their mom and one from their dad. And in their case, they have 1 normal functioning copy, which I'm going to call capital F for that FXN gene, and 1 abnormal copy, which has too many of that GAA repeat, which I'm going to call lowercase f. Even though this person carries an abnormal copy of the gene, they're still going to be asymptomatic, not going to have any problems with Friedreich's ataxia because their normal copy of the gene is going to be creating plenty of that good protein. Even though the abnormal copy isn't going to be creating much protein, you're still overall going to be creating enough. All right, but here's where the issue comes in with recessive genetic illnesses like this. We have this couple here who are both carriers in that they have 1 capital F and 1 lowercase f of a normal and an abnormal copy of this gene. Well, each of their kids is randomly going to receive a copy of this gene from each of their parents. Let's say this one here receives normal F from mom and also normal F from dad. They're not going to be a carrier at all. They're going to have both copies of that normal gene. This kid here is going to get normal F from mom and lower F from dad. And they're also going to be a carrier, meaning that they're going to be asymptomatic but still have a copy of that gene themselves. Next kid is going to have lower F from mom and normal large case F from dad. They're also going to be a carrier as well. And our last one is going to unfortunately end up with the recessive lowercase f, the abnormal gene from both parents. It's completely random which copy you get from which parent, so it's by no means a guarantee that you're going to end up with exactly this ratio of which kids are going to get what genes. It's totally possible to have 10 kids who all end up with the normal function gene, or 10 kids that have always the abnormal function version. It's just statistically likely that it's going to come out in approximately this ratio. Let's look at a different circumstance where we have, say, mom here with both copies of the normal functioning gene, and dad here who's a carrier with only one copy of the abnormal. Neither of them are going to be symptomatic, and on average, 2 of their kids are going to end up with 0 copies of that abnormal gene, and 2 of their kids are going to end up being carriers, but with very few exceptions of a random mutation, basically none of their kids are ever going to be symptomatic. This type of issue is often why we end up talking to genetic counselors for our patients, so that they can talk about, oh, what are the odds that, say, an uncle or a cousin or my kids might end up being affected by this disease as well, and help us with family planning. It's also important to keep in mind that for reasons that I'm not going to go into today, this GAA repeat, whenever it's inherited, tends to get a little bit bigger. Let's say mom has only 90 copies of that GAA repeat within her abnormal gene. Well, one of her kids might end up with, say, 120 copies. It's just part of the process of when it gets passed down, it gets a little bit longer. And unfortunately, as we talked about before, it tends to become more serious and symptomatic in terms of creating less of that protein with each generation down it goes. Yet another reason it's important to discuss these things with a genetic counselor. So I've talked a lot about how that protein frataxin is made and how not having it can cause various issues, but I still really haven't given you a great answer for what frataxin itself does. And that's because, to be completely frank, we don't have a 100% clear answer for that yet. Is it somehow involved in energy regulation? Well, tissues that require the most energy seem to be the most affected, so there's some thought in that area. Does it play a role as an antioxidant? Well, it seems like if you increase the amount of frataxin in certain animal models, that they have greater antioxidant capability, which is basically a way of saying that you have greater tolerance for having chemicals in your bodies that could cause damage but are sort of buffered out by having antioxidants. It's also thought that it definitely has some sort of role in iron storage and involved in these iron-sulfur molecules, but what exactly that has to do with causing symptoms and why not having it creates those symptoms is up for a little bit of debate. There have even been some research trials in terms of medications that affect the amount of iron you have in your body and seeing if that could potentially reduce symptoms, and unfortunately those didn't really show to have a very good outcome. And finally, why some tissues are affected more than others? Well, there's lots of tissues in the body that require a lot of energy. I know I said that the heart and the brain and the other parts of the nervous system tend to be affected a lot, as well as the pancreas, but they're not the only ones that require a lot of energy. So it's not really 100% clear why frataxin seems to be so critical in some of these tissues and not in others. What is clear though is that ongoing research could potentially provide some answers for this, especially if we focus on the genetic aspect of it, where if we can simply repair the function of the gene, we don't even necessarily need to know what frataxin does, so long as we can help the body create more of it the way it should.